Okay, so this might be a slightly longer video. We're going to go through the first 3 steps of cellular respiration, which is glycolysis, the transition, and the citric acid cycle. So we're going to start off with glucose. Now remember, glucose is a molecule made of 6 carbon atoms, and that's what these 6 black markers represent. 1, 2, 3, 4, 5, 6 carbons. And they're joined to each other by covalent bonds, which are really electron pairs. And each of these red linkages represents the bonds joining these carbon atoms together. They're high energy electrons that are gluing this molecule together. Now remember, in real life, glucose is a ring, a hexagon kind of looking thing. I can't make this a hexagon, so we're just going to go with a straight chain. So we start off with glucose. Now our first step in cellular respiration is called glycolysis, and it basically means glycolysis, the lysis, the breaking of sugar. So that's what the word really means, and it happens in the cytoplasm. My setup is kind of not like the lecture room. In the cytoplasm. All right, so what happens in glycolysis? We're going to take our 6 carbon glucose and we're going to do this to it. Break it in half. We did glycolysis. We split the sugar. All right, so at the end of glycolysis, we end up with 2 molecules. And each one of these is called pyruvic acid or pyruvate, and it has 3 carbons in it. All right, so I'm going to write over here that glucose is going to turn into pyruvic acid. Sometimes it's called pyruvate. It's the same thing. And this is 3 carbons all hooked together. See? 1, 2, 3. All right, glucose is 6 carbons. In fact, the actual equation is C6H12O6, right? All right. So that's really the kind of overall chemistry of it here. But there's one more thing we have to do. When we take our sugar, which is what we start with, glucose, and we do glycolysis, break it in half, because we've broken a bond here, we can harvest these electrons that were originally holding this together. So I've pulled some high energy electrons out of this molecule when I split it. And we're going to use these later, but right now we need to put them somewhere special, somewhere safe, and get them to where we need them. So we're going to use this. This molecule is called NAD. It's an electron carrier. I like to think of it as a taxi, which is why it looks like a taxi, kind of. And we're going to put this high energy electron in the taxi. Right now the taxi is empty, so it's called NAD. When it gets the electron in it, it turns into NADH. So NADH is the full taxi, the taxi with passengers. This is the empty taxi, how it starts off. So I'm going to do this whole thing one last time for glycolysis. All right, so we're going to start with glucose. We're going to break it in half by breaking that bond, harvest the electron, bring along an empty taxi, put the electron in the taxi, and now the taxi is called NADH. There's the electron. I'm going to stash that for later because we're going to need it. And what I have here is 2 molecules of pyruvic acid or pyruvate. That's glycolysis.